Now we're going to look at enzyme inhibition and regulation. One of the things that enzymes can experience is a decrease of activity because of chemical means. An enzyme inhibitor is a substance that decreases the activity of an enzyme. Enzyme inhibitors can be irreversible. And when we have irreversible inhibitors, they're going to form a covalent bond within a specific functional group within the enzyme that basically makes the enzyme be inactive. One example of an irreversible inhibitor is going to be heavy metal poisoning. Examples of heavy metals could be mercury or lead. Mercury and lead specifically bind covalently to cysteine residues, and we can observe that now by having the thiol groups of the cysteines and covalently binding it with mercury, that will result in the enzyme being damaged and being inactivated. Both mercury and lead poisoning may cause neurological damage if they left untreated. When it comes to irreversible inhibitors, not all of them can act as poisons. As a matter of fact, some of them are actually useful as therapeutic agents. One example of this is the use of antibiotics. An antibiotic is a substance produced by one organism that kills or inhibits the growth of another organism. Ampicillin, which is a specific type of penicillin, is such example. Specifically, penicillins are used to treat different infections that are actually created by bacteria. And when this molecule was generated and used in medicine, it actually revolutionized medicine because they found a way in order to now inhibit the growth and actually kill the bacteria that were creating illnesses like pneumonia, strep throat, or even scarlet fever. Okay. So understand that antibiotics are actually useful and they happen to be irreversible inhibitors. Another type of enzyme inhibitor is going to be a reversible inhibitor. Reversible inhibitors bind reversibly to an enzyme. When the enzyme binds this reversible inhibitor, it can do it in two different modes, competitively and non-competitively. Let's look at the difference between a competitive inhibitor versus a non-competitive inhibitor. A competitive inhibitor is going to be a molecule that competes with the substrate of the enzyme for the active site of the enzyme. Interestingly, competitive inhibitors can be reversed by increasing the concentration of the substrate for the enzyme. If enough substrate is added, it eventually displaces the bound inhibitor. As you can see in figure 12.35, if an enzyme has its regular substrate, a competitive inhibitor has a shape that it is similar to the substrate and it can bind at the active site. Let me show you an example of a competitive inhibitor by looking at another type of antibiotic. So this type of antibiotic is known as sulfonylamide, and it is used to treat bacterial infections. The enzyme dihydropteroate synthase has a regular substrate that is known as para-aminobenzoic acid. Sulfonylamines are similar in structure to para-aminobenzoic acids. The acronym for para-aminobenzoic acid is PABA. Sulfonylamines are similar to the regular substrate for the enzyme, in which we have a benzene ring with an amino group. In PABA, we can see a carboxylic acid. But in the sulfonylamide, what we see is a sulfur with two double bonds to oxygen connected to an NH2 group. Since these molecules, at least in this region, they're similar, sulfonylamide can be a competitive inhibitor with PABA for specifically treatment for bacterial infections. If inhibitors don't bind the active site, and it binds somewhere else in the enzyme away from the active site, that is known as a non-competitive inhibitor. When a non-competitive inhibitor binds away from the active site, that means that it binds allosterically. The interaction between the enzyme and the non-competitive inhibitors causes a three-dimensional shape change on the enzyme, and that's how the reduction of the activity occurs. Understand that this conformational change that happens with non-competitive inhibitors, since it affects the active site, that's where the inhibition comes from. When the active site change has occurred because of the presence of a non-competitive inhibitor at the enzyme, understand that the substrate cannot properly bind the enzyme anymore. Now, different from competitive inhibitors, non-competitive inhibitors, they need to be reversed specifically by chemical means, not by the addition of additional substrate. We can see an example of a non-competitive inhibitor by looking at DNP. DNP is 2,4-dinitrophenol. Specifically, DNP is going to affect a specific area of the cell that is called the mitochondria, which is where that energy molecule ATP is generated. So understand that originally DNP was utilized for inhibiting a specific enzyme in the mitochondria, and that was marketed at first in the 1930s as a weight loss drug, but it was banned because it actually create toxicity in the body. You can see that in this flow chart, a summary of the types of inhibitors that you need to be familiar with in terms of the definitions. We're going to have inhibitors that are irreversible. And irreversible inhibitors, remember that they are going to covalently bind with the enzyme. Reversible inhibitors, on the other hand, they can be reversed. If they bind the active site, they're known as competitive inhibitors. But if they bind allosterically, meaning in a site other than the active site, that is known as a non-competitive inhibitor. The difference between the reversibility of competitive and non-competitive is that in competitive inhibitors, you can add more substrate to reverse them. But in non-competitive inhibitors, you need chemical means, meaning a chemical reaction needs to be added to the enzyme that contains the non-competitive inhibitor in order to reverse its effect. Enzymes can also be controlled and regulated by what is called allosteric regulation. We kind of see some of this when we were looking specifically at non-competitive inhibitors. Allosteric regulation basically means that is going to be when a substance binds to an enzyme at a location other than the active site, and it actually alters the catalytic activity for that enzyme. So understand that an example of that was the use of a non-competitive inhibitor. The other type of regulation that can happen in enzymes is actually feedback inhibition. In feedback inhibition, this is a process in which the end product of a sequence of enzyme catalyzed reactions inhibits an earlier step in the process. To look specifically at an allosteric regulation that additionally is also an example of feedback inhibition, here we have the specific enzyme catalyzed reaction for the conversion of the amino acid threonine all the way to isoleucine. You can see that the conversion of threonine to isoleucine, actually, it is performed by 5 different enzymes. And the end product, which is isoleucine, in turn, once is produced, it can actually go back and inhibit the very first enzyme in the pathway. By doing this, if you have produced enough isoleucine in a living organism, now instead of putting more threonine down the pipeline to produce isoleucine, you can block the production of isoleucine by blocking the first conversion of threonine down the pipeline to make isoleucine. So in this feedback inhibition, we can see how isoleucine is specifically an inhibitor of the first reaction within the conversion of threonine to isoleucine.